Chemotactic microdroplets for targeted drug delivery is an emerging technology that could transform medicine, making drug treatments more precise, efficient, and personalized. Have you ever wondered why some medications have serious side effects, or why certain drugs struggle to reach the exact spot in the body where they are needed? This challenge has led scientists to explore chemotactic microdroplets, which are tiny liquid droplets that can move autonomously toward specific chemical signals, much like how immune cells navigate to infections in the body. Chemotaxis is the ability of biological cells, bacteria, or synthetic particles to move in response to chemical gradients. For example, immune cells can detect chemical signals from bacteria and move toward the infection site to fight off pathogens. Researchers are now applying this concept to synthetic microdroplets, designing them to detect and follow chemical cues to deliver drugs precisely to diseased tissues. This means instead of flooding the entire body with a medication, which can cause unwanted side effects, these microdroplets could self-propel directly to the affected site, reducing damage to healthy tissues. Why is this important? Think about cancer treatment. Traditional chemotherapy drugs spread throughout the body, killing both cancerous and healthy cells, leading to hair loss, nausea, and weakened immunity. But chemotactic microdroplets could be programmed to seek out tumor sites, releasing the drug only when they reach cancerous cells. This would increase treatment effectiveness while drastically reducing side effects. Another exciting application is in neurological diseases, where delivering drugs to the brain is challenging due to the blood-brain barrier. Scientists are exploring microdroplets that can sense inflammation or specific neuron signals, helping them navigate directly to damaged nerve cells in diseases like Alzheimer's or Parkinson's. Could this technology one day enable smart treatments for brain disorders without the need for invasive procedures? In infectious diseases, chemotactic microdroplets could act like artificial immune cells, traveling through the bloodstream to locate bacterial infections and deliver antibiotics exactly where they are needed. This could help fight antibiotic-resistant bacteria, as the drugs would be concentrated at the infection site rather than spread throughout the body, reducing the risk of resistance development. How do these microdroplets move? Scientists use special biochemical coatings, surfactants, or enzyme-driven reactions that allow the droplets to respond to chemical gradients. Some are even developing microdroplets that can be magnetically controlled, providing an external way to guide them through the body. Researchers are also investigating whether these microdroplets could sense and react to real-time changes in the body's chemistry, adapting their behavior dynamically. Despite its promise, challenges remain. Ensuring that these microdroplets can be safely eliminated from the body after delivering their payload is crucial. Scientists are working on making them biodegradable so they naturally break down after use. Additionally, scaling up production and ensuring regulatory approval will be necessary before this technology can be widely used in medicine. The future of targeted drug delivery may lie in chemotactic microdroplets, offering more precise, less invasive, and highly effective treatments for cancer, infections, and neurological disorders. Could we one day see a world where smart, self-navigating drug carriers eliminate diseases with minimal side effects? The research is advancing rapidly, bringing us closer to that reality. What other medical challenges do you think this technology could help solve? Chemotactic microdroplets are an emerging approach in targeted drug delivery that harness the ability of microdroplets to navigate chemical gradients in biological environments. Inspired by natural chemotaxis, the movement of cells and microorganisms in response to chemical stimuli, these synthetic microdroplets can autonomously move towards specific tissues or disease sites by incorporating biocompatible polymers, nanopolymers, or active molecules. Researchers design microdroplets capable of responding to pH, inflammation markers, or specific biomolecules. This technology represents a step toward intelligent drug delivery systems that optimize treatment while minimizing systemic side effects. Chemotactic microdroplets engineered with pH responsive properties have been developed to deliver anti-cancer drugs directly to tumor sites, reducing toxicity to healthy tissues. These microdroplets act like heat-seeking missiles, homing in on disease sites and delivering payloads with precision. The movement of chemotactic microdroplets is driven by physiochemical interactions with surrounding fluids, enabling them to follow gradients of specific chemical signals. This behavior is often achieved using surface tension gradients, interfacial reactions, or Marangoni flows, phenomena where differences in surface energy drive directed movement. Additionally, microdroplets can be loaded with enzymes that catalyze reactions, producing propulsion forces that direct them toward target tissues. These mechanisms mimic biological processes such as how immune cells detect infection by following cytokine gradients. Microdroplets infused with catalase, an enzyme that decomposes hydrogen peroxide, have been used to create self-propelling drug carriers capable of navigating inflamed tissue regions. Chemotactic microdroplets behave like scent-tracking dogs, following chemical trails to their intended destinations. The composition of chemotactic microdroplets is crucial for their stability, responsiveness, and biocompatibility. These microdroplets are typically made of biocompatible oils, hydrogels, or polymers capable of encapsulating therapeutic agents. Functionalized nanoparticles, or surfactants, are often integrated into their structure to enhance mobility and responsiveness. Smart materials, such as stimuli-expressing lipids or microgels, enable controlled release of drugs upon reaching the target site. Advances in microfluidics allow precise fabrication of microdroplets with tunable properties for different biomedical applications. Lipid-based microdroplets loaded with chemotherapy drugs have been modified with folic acid receptors to enhance targeting of folic-expressing cancer cells. Choosing the right materials for microdroplets is like designing an autonomous drone. Each component must be optimized for navigation, endurance, and mission success. Tumor tissues create unique chemical microenvironments characterized by acidic pH, hypoxia, and elevated levels of inflammatory molecules. Chemotactic microdroplets can be engineered to exploit these differences using pH-responsive polymers or oxygen-sensing materials to guide their movement toward cancerous regions. Once in proximity, they can release their drug payload in response to specific environmental triggers, ensuring localized treatment while reducing off-target effects. Researchers have developed microdroplets that migrate toward lactic acid-rich tumor regions, releasing doxorubicin upon detecting acidic conditions. These microdroplets function like homing pigeons, navigating toward their programmed location based on environmental cues. Microfluidics plays a critical role in the fabrication of chemotactic microdroplets, allowing precise control over size, shape, and composition. Microfluidic devices enable the generation of uniform, monodispersed droplets with specific chemical properties tailored for drug delivery. This approach also allows for the encapsulation of multiple therapeutic agents. Additionally, microfluidic synthesis ensures reproducibility and scalability, making these technologies more viable for clinical applications. Microfluidic techniques have been used to create Janus droplets, microdroplets with two distinct compartments, allowing for dual drug release in chemotherapy. Microfluidic fabrication is like a precision assembly line, ensuring each microdroplet is manufactured to exact specifications for optimal performance. Chemotactic microdroplets can be designed to release drugs in response to environmental stimuli such as temperature, pH, or enzyme activity. By incorporating smart polymers that degrade or swell under specific conditions, these systems ensure that drug delivery occurs only at the target site. This strategy improves therapeutic outcomes while reducing the risk of premature drug release. Thermosensitive microdroplets have been engineered to release antibiotics when they reach inflamed tissues, which are typically warmer than normal body temperature. These microdroplets act like temperature-activated coffee cups, releasing their contents only when the right conditions are met. Despite their promise, chemotactic microdroplets face challenges in clinical translation, including immune system interactions, stability in biological fluids, and large-scale production. Ensuring biocompatibility while maintaining chemotactic behavior is crucial for in vivo applications. Additionally, controlling their movement in complex physiological environments requires further refinement of chemical sensing and propulsion mechanisms. Ongoing research focuses on integrating bio-inspired navigation strategies to improve their effectiveness in real-world settings. Current studies are exploring microdroplets coated with immune-evasive materials to prevent rapid clearance from the bloodstream, extending circulation time. Bringing chemotactic microdroplets to clinics is like adapting robotic rovers for Mars exploration. Every environmental challenge requires a tailored solution. The future of chemotactic microdroplets lies in integrating artificial intelligence, AI, and nanotechnology to enhance targeting precision. AI-driven predictive models can optimize microdroplet formulations based on patient-specific data, personalizing treatment strategies. Additionally, hybrid systems combining chemotactic microdroplets with nanocarriers could enable multi-stage drug delivery, ensuring sequential release at different disease progression stages. These innovations hold promise for revolutionizing precision medicine. AI-powered simulations are being used to design microdroplet systems that dynamically adjust their chemotactic behavior based on real-time physiological conditions. Advancing chemotactic microdroplet technology is like refining self-driving cars. Leveraging AI to make autonomous navigation safer and more efficient. As chemotactic microdroplets enter biomedical applications, ethical and regulatory considerations must be addressed. Ensuring patient safety, long-term biocompatibility, and potential unintended effects requires rigorous testing and regulatory approval. Additionally, ethical concerns about using AI-driven drug delivery systems highlight the need for transparent validation processes and patient consent frameworks. Developing standardized guidelines will be essential for their integration into mainstream healthcare. Regulatory agencies are beginning to establish safety protocols for microdroplet-based therapies, requiring extensive preclinical testing before human trials. Navigating regulatory approval for chemotactic microdroplets is like piloting an experimental aircraft. Rigorous safety checks must be passed before widespread use.